Hello everyone, I'm Yuqing. I'm from the Division of Quantitative and Method, Office of Research and Standard under the Office of Generic Drugs. So before I get started, I would like to thank to the meeting organizers to invite me for this presentation. So today I will talk the considerations for FEV1-based comparative clinical endpoint or pharmacodynamic bioequivalence studies for orally inhaled drug products. So this is my disclaimer. This presentation represents the views and perspectives of the speaker and does not necessarily reflect the views of the US FDA. So in today's talk, firstly, I will present the challenges for FEV1-based comparative clinical endpoint or pharmacodynamic bioequivalence studies for locally acting orally inhaled drug products. So those challenges includes the high variabilities, the flat dose-response relationship, and to provide a measure of those challenges, I'll present the data that we analyzed using the FDA received data. And then I will discuss the potential opportunities and strategies to address those challenges, including the covariate adjustment and the considerations for alternative BE studies. And I have the summary and the references at the end of my slides. So FDA currently is using a weight of evidence approach to demonstrate a generic locally acting orally inhaled drug product. It's equivalent to its R&D. So this method generally includes the in vitro studies and formulation sameness and device similarities to provide the equivalence in product performance, and a pharmacokinetics BE studies to provide the equivalence in systemic exposure, and a comparative clinical endpoint or pharmacodynamic BE studies to provide equivalence in local drug delivery. So taken together to provide the bioequivalence for the oral inhaled drug products. So FDA is continuously publishing PSGs. So based on the currently published PSGs, the baseline adjusted mean FEV1 AUEC, such as AUEC at 12 hours, AUEC 24 hours on the first day, or the trough AUEC on the last day are recommended in majority of the orally inhaled drug products as the comparative clinical endpoint studies, including the inhaled corticosteroids, the LABA, and the SABA products. So those studies are commonly used as parallel design over crossover design because of the study duration. Some of the study may take 4 to 6 weeks. So for products that contains the short-acting beta-2 agonists, like albuterol and levoalbuterol products, the PDBE study with dose scale analysis is recommended. So for albuterol products, a bronchoprovocation study is recommended with the endpoints PC20 and PD20, just as Key just mentioned. And for demonstrate the PDBE studies, an adequate dose-response relationship is needed. So those are some challenges in comparative clinical endpoint or PDBE studies for the orally inhaled drug products. So firstly, for those studies, they often need to conduct in patients. And then the FEV1, it can be influenced by many intrinsic and extrinsic factors, such as age, sex, height, genetic variations, and even the disease progressions. And they are observed the high day-to-day variations in FEV1 that has been reported in patients. And there are high variabilities in FEV1, so lead to the requirement of large sample size for the bioequivalence studies. So this is results that FDA analyzed based on the studies based on the studies submitted to the FDA, and this results has been published in CPT last year. I have listed the full details of this publication, the name, in the reference list, and you're welcome to check the reference list and read the full details. So basically, in this study, we collected the study that's submitted to the FDA that contains the comparative clinical endpoint study with the FEV1 endpoints. And we analyzed the variabilities from those studies, basically the CV percentage. And we found that the mean variabilities, the CV percentage of FEV1 AUEC and the trough FEV1 were more than 100 for both the test products and the reference listed drugs. And we also found the variabilities were over 200 for the placebo groups. So part of the reason we are seeing such a high variability, it could be because of the flat dose response relationship and because the study is conducted in patients. So this is the predicted sample size based on the variabilities we just seen. And we are taking from one example data set to perform the simulations and to calculate the passing rate based on those based on the TR ratio from that example data set and based on the variabilities from that example data set. So here the curve you can see, the red curve is to represent the trough FEV1, and the blue curve is the BE based on the AUEC. So from this simulation, we have seen a sample size of around 750 for both TEST and R&D groups is needed to achieve 80% study power for BE2 conducted based on FEV1 AUEC. And a sample size of around 1,400 is needed to achieve 80% study power for the BE based on the trough FEV1. So you can see this, it needs a very large sample size. So as I just mentioned, part of the reason is because this dose-response relationship is quite flat. And for most of the drugs that with the approved dose, they are on the flat region of the dose-response relationship, the response curve. And we have studied several, multiple drugs that are listed here. So we are trying to see the dose-response relationship here. So for majority of the drugs that we studied, you can see from the table here. So majority of the drug has no significant dose-response relationships, or the approved dose is on the plateau of the dose-response curve. And we have seen for formoterol, they appear to have a dose-response relationship on PC20, but still there's no strong evidence to support a PDB study can be recommended for this product. So the current investigation did not find a very strong evidence to recommend a PDBE studies for the products that listed here in lieu of the comparative clinical endpoint studies. So now let's look at some potential opportunities to address those challenges. So the first one I listed here is a covariate adjustment. So the covariates can be used to adjust the baseline variables that estimating the treatment effects for the comparative clinical endpoint studies. And for covariate adjustment, it often been used in parallel group trials. And the baseline covariates can be some demographic factors and can be the disease characteristics or other related study informations. I'm putting one case example we did regarding the covariate adjustment. So this is from one example data set. So we used the ANCOVA analysis on this example data set, and we analyzed all the possible covariates. Here, the covariates we analyzed in this specific dataset includes the demographic variables, age, sex, weight, and height, and the study informations, the study region, and other data like baseline FEV1 value, baseline FVC values. So we conducted the covariate selections for all those potential covariates, and we conducted using a stepwise regression and to minimize the AIC values. So based on this example data set, the selected ANCOVA models, it can explain about 80% the variabilities for the baseline FEV1, but only a limited portion for the variability, like 8 to 20% for the primary endpoints for FEV1, AUEC, and the trough FEV1. And for this example data set, we find the use of ANCOVA with selected covariates can reduce the number of subjects approximately about 10%. So that being said, if you have to recruit like 1,000 subjects, now it can reduce to 900 subjects. So for this method, the covariates adjustment, the prospective applicant should pre-specify the ANCOVA models with the prior knowledge to like which covariates can be the potential one, and in their statistical analysis plan before conducting the comparative clinical endpoint BE study. Of course, the scientific community, including the FDA scientists, are trying to find if there are alternative PD models or endpoint that can be a more sensitive endpoint compared with the currently recommended FEV1 endpoint. So we have looked into the literatures and some studies. So there are some alternative PD models were available in the literatures. For example, in the case of ICS, the inhaled corticosteroids, several alternative PD models were available, including the induced allergy challenge, asthma stability models. But many of those PD models are still challenging to be used for the BE assessment due to the highly variable dose-response relationship. So, however, FDA encourages to explore those novel approaches or BE matrix that may offer improved study sensitivity in establishing BE for the already inhaled drug products. I would like also to mention currently FDA is also recommending the alternative approaches in lieu of the clinical comparative endpoint study or PDBE study. This can be also an alternative options too. So the PSGs for beclomethasone MDI suggested the alternative approach may include, but not limited to, the following studies, such as the additional in vitro testing, like a more predictive ASPD testing, some dissolution testing, and immediate sprays. And also it can include the alternative, like additional in vivo PKBE studies. And it can also include the quantitative methods and modeling, such as the CFD models and PBPK modelings. So overall, the goal of the alternative approach, it should provide the scientific justifications to help ensure the equivalence of the product at the local site of the action in the lungs. But this is what the clinical endpoint study is representing, the local site of action in lungs. And you can find the details in the FDA PSGs for beclomethasone MDI. So as already mentioned by Rob and Key, the early communication with FDA, it strongly encourages if you are plan to do the alternative approaches, and it can be via the pre-ANDA meeting pathway. And early communication with the FDA can help to clarify expectations in product development and to assist the applicant to submit an ANDA as complete as possible. So here are some take-home messages from my talk. Firstly, the FEV1-based matrix are the most recommended in the comparative clinical endpoint for the locally acting orally inhaled drug products. And we have seen there are high variabilities for FEV1-based matrix, resulting in a need of a very high sample size, maybe more than 1,000, to achieve sufficient power to establish the BE for the generic orally inhaled drug product. And we also have seen the flat dose response relationship that may lead to the low sensitivity in detecting clinical meaningful differences and consequently the formulation related differences for the drug exposure between the test and reference product at the site of action. So there are potential opportunities, for example, the one case that I presented using the covariate analysis to reduce the sample size and to explain some of the variabilities in the study. And FDA continues to explore and evaluate new approaches for BE to help mitigate the high cost and the time investment associated with the development of the locally acting generic orally inhaled drug products. So prospective applicants and academia are encouraged to participate in this effort by submitting the new approaches in the product development via the pre-ANDA meeting, and other academia can advance the research in collaborative grants and contracts with the FDA. Here are the references listed that I discussed today, and the first one that listed here is the one contains the results that I presented today. And I'd like to acknowledge the people listed here, especially the first one, Jiang, who is the primary author for the paper that I just discussed, and all the co-authors that listed here. And I'm happy to accept the questions, and I'll back in the panel to address the questions. Thank you.
As my slides come up, and hopefully the clicker will work now. I just wanted to thank everyone and Parmjeet for the introduction earlier on. My wife also wishes to thank everyone that we're finally having face-to-face symposiums again. It's not only been 3 years since I've travelled anywhere on business out of the country, it's been 3 years since I've left my home office, so she's delighted to be rid of me for a few days. This is a really kind of important topic to me. It's been 12 years of my career, maybe a little bit more. I say in my biography, I'm a respiratory lifer, and I've worked in inhaled products all my career. And as was mentioned earlier, I started in innovator companies and then became a poacher and worked my way to generics companies later on in my career. When I looked at the topic for the symposium today, it was reviewing successes. As Robert mentioned earlier on, there aren't a great deal of successes so far in terms of developing inhaled products as generics in the US market. But as Robert also pointed out, it's been an infinite increase since 2018. And so with the aim of reviewing successful implementation of one of these clinical endpoint bioequivalent studies, I wanted to use our experience at Viatris with development of Wixela Hub, talk a little bit about that. I thought I'd better start because for those outside the US, you may not know much about Wixela Hub. So this was approved by FDA in 2019 as a substitutable generic equivalent to Advair Diskus. I like to think of it as a complex generic with added complexity. It felt like complexity squared working on this within industry. And it was the first one filed to my knowledge for ANDA and the first one approved. Why was it so complex? It was a powder, a dry powder inhaler. It was a combination product of 2 APIs, 3 product strengths. And it included both the LABA, which was challenging enough in itself, but also an inhaled corticosteroid. And for reasons that have already been discussed around dose response, inhaled corticosteroids were a very difficult thing to be developing, let alone in combination. And also, in the picture up there in the grey, it was a novel device. So it was comparable in many aspects to the discus inhaler. Number of doses it contained, lots of patient device interface, how the devices were contained within the devices. But it was a novel device and it wasn't identical, and in particular the plane in which the device was used was different. Since that experience, almost 5 years on, I've also had the experience of working with the team at 3M and Kindiva, who are our partners for Brainer, which is a generic equivalent of the Symbicort HFA MDI, which was approved by FDA last year. So 5 years apart, I've had experience with 2 clinical endpoint bioequivalent studies, one the first to go through and one a subsequent entry into the approval pathway. And I've seen how that approval pathway has improved over those 5 years. And we learned a lot along the way, and I believe the agency has done as well. And, you know, whilst I'm not talking about that today because it was mainly developed by Kindiva, it's in my mind, and if anyone has questions about it, then I'm happy to take whatever I can later on. I think to be able to review the success, you really need to start back at where we started from to realise how far we've come. And you'll notice in my first slide, I mentioned 2011. That was when I started with Mylan, who became Viatris. But actually, in my second and third slides, I think it mentions 2009, 2008, 2007 even, when I was working with Pfizer, which is where this product kind of originated. And there was no guidance at that time. I'll come on to that in a minute. Then in 2013, with lots of people, both at the agency and in industry, trying to come up with novel methods that could be used in terms of local equivalents of these products. Finally, guidance was produced by the agency, which gave us a pathway to approval for this product. Until that point, industry had been investing at risk of there not being a pathway to approval for this type of product. And that's a big ask when the development programs are as expensive and complex as the ones required for this product. I'm then going to go on to describe the study itself and show you the results of the study, and then talk about some of the strengths, the weaknesses, many of which have already been touched on, and some of the learnings which I took from it. And then really just finish the discussion, finish the presentation with a couple of slides at the end saying, is this weight of evidence, is it appropriate? And I was really pleased to hear Robert describing earlier on, you know, that there is an understanding that this is a heavy piece of information in that weight of evidence approach, which perhaps doesn't generate as much assurance of equivalence as much less heavy pieces of evidence in that weight of evidence approach. And I'll come back to what I mean by that later on. So teeing up then the rest of the workshop, which is about alternatives, and we all look forward to alternatives. So when we started in 2009, as I mentioned, there were no generic high complexity orally inhaled products approved in the US. There were nebules approved, and going down this route, there had been short-acting beta-agonist albuterol MDI products, which at that time were the CFC products, which came in as generic equivalents, I think in the late 80s, and subsequently had been withdrawn. So we didn't really have any precedents with which to start out on our pathway. And at the time, there were no product-specific guidances. and there was no formal FDA guidance about these products and how you would get them there. There were, of course, plenty of presentations and, of course, workshops. And, I think what we're seeing today is an iteration of workshops which started back in the late noughties, which have developed to get us to a place where we can get products through registration now. And now we're seeing a new iteration, which is around making that pathway as efficient as possible to try and encourage that metric around 4 out of 33 to become a much larger number. The weight of evidence approach was already mentioned in publications and had been applied, I guess, to some degree to the albuterol MDI products developed years before. So we knew a bit about Q1, Q2 formulation, comparability, in vitro equivalence to a degree. We knew that you needed systemic pharmacokinetic bioequivalence, but we didn't know any of the detail of that. We didn't know whether it needed to be for every strength, what metrics we were going to look at, what acceptance criteria would be applied. Would that be enough? Would you need to have systemic pharmacodynamic studies as well? So we knew the general way that the agency were moving here, but we didn't know any of the detail of it. Local equivalence to me was always the scariest thing. How were we going to determine local equivalence within the lung? And conceptually, I found this the most challenging part of defining a strategy. And obviously the reason it's required was mentioned earlier on, because these are topical products and pharmacokinetics is reflecting distribution from the site of action rather than to the site of action. I'll come back to that towards the end of my presentation. The targets for local equivalence were high, and I just pulled out a slide there from, I think, an RDD presentation that Wally Adams gave in 2009, 2010. But in much the same way we heard about earlier on, the PD models for SABA, the same kind of terms were being used to describe what we could try and do for ICS and for LABA. And so industry weren't the only ones having to deal with this. FDA were also sponsoring their own research at the time. I just pulled out there the asthma stability model with Richard Ahrens in Iowa, and looking at exhaled nitric oxide as potential pharmacodynamic markers in which were being examined around dose response of inhaled corticosteroids at the time. However, the kind of lack of a defined pathway to registration meant that some large generics players decided this just wasn't an area which they could participate in. And as a result there, the bottom press cutting, we were in 2010 with patents expiring in 2011, and Glaxo were, feeling very comfortable that there was no one who was going to be turning up anytime soon to try and take US market share from them. So against that backdrop, we were considering the program ourselves, and we had what they were called scientific advice meetings with OGD and a division back in 2009, 2010, in which we presented the product concept and our thoughts around development of it. OGD definitely led on those meetings, but Badrul Chowdhury was in position at the division in those days, and he was a part of that as well. And we left clear that Wixella Inhub was comparable enough to Advair Discus that an ANDA was going to be our route to approval, which left us even more scared that without this clinical equivalence piece nailed, we had a pathway, we had a product which needed registration via a pathway which didn't really exist at that time. So against that backdrop, we started off looking at these different models that were available, and we selected one for the salmeterol component, which unsurprisingly is the methacholine challenge model, which has been talked about a lot this morning for albuterol. And we wondered whether this could be used to look at salmeterol as well. And obviously critical in that would be, did you see a dose response? How steep was that dose response? And excuse the size of this, what that shows is 2 different doses of salmeterol from within Advair, the 50 dose, and then 2 puffs of that, the 100 dose. And this is looking 1 hour after inhalation and seeing very little dose response between the low and high dose in terms of methacholine PC20, suggesting to us that whilst this model had proved useful for albuterol and may prove useful for formoterol, as has been highlighted earlier on, it wasn't suitable for salmeterol in Advair. We also ran another, and I would say these were well-run, well-controlled studies looking at exhaled nitric oxide, and in this case looking at the FP component of Advair discus, and looking at a range of doses, not only the registered doses of 50, 150, 250 and 50, 500 twice daily, but we also looked at a very low dose, a once daily dosing of the 50, 100 strength of Advair. We looked across 14 days of dosing. We did a crossover study. We ensured good washout between periods. But what we found was that all of the dose response for exhaled nitric oxide as an endpoint fell between the once daily dosing of the low strength and the twice daily dosing of the low strength. And around about the same time, FDA completed their own study where they looked at fluticasone in the MDI product, and also found a lack of dose response. They found, as we did, that exhaled nitric oxide was simply too sensitive to the inhaled corticosteroid component. So we were left at the end of middle of 2013 with 2 method studies, both well executed, both of which gave information which suggested these methods were not useful in terms of showing local bioequivalence of the salmeterol and fluticasone components within Advair discus, let alone the generic that we were developing. And we were ready to take our position back. In fact, we'd sent data from both of these studies into FDA, and we were ready to go back in August 2013 to talk about how are we going to get this product to approval in terms of local clinical equivalence. We were put off by a month. The agency said, just maybe come in September. And in September 2013, the product-specific guidance, based on the totality of the agency's experience, I'd like to think some of which was our experience as well at the time, the product-specific guidance was issued for salmeterol fluticasone oral inhalation powder. And crucially, it included details of a clinical endpoint bioequivalence study, which we felt could be executed. And that's what I'm going to just talk about now in the next few slides. So as you've heard already, it was a randomized, and actually we ran this as a double-blind, double-dummy study. We were able to access placebo products for discus. So it was a fully double-blind, double-dummy study, which created its own problems I'll come back to maybe in terms of learning. Parallel group study, 3 to 4 week, single blind placebo run-in period, 28 days treatment. We conducted it only in adults over 18 years of age, and we chose an FEV1 range to begin with of 60 to 80% of predicted. We were surprised how many patients arrived at clinic with lower lung functions who were not taking any kind of asthma maintenance treatment. And this study was entirely conducted within the US. And so actually we amended the protocol during the study to reduce that lower limit down to 50% of predicted, because those were the patients who were coming in and were able to participate in the study. The treatments, obviously, the lowest approved dose of Advair discus, it's equivalent of in Wixella Inhub and a placebo, and we used a 5:5:1 ratio. The endpoints we've already talked about, and the reason for the 5:5:1 ratio is the assay sensitivity, showing that Wixella and Advair were both significantly different to placebo. For both endpoints is a rather simple thing to demonstrate, requiring not many patients, whereas equivalence testing showing that the ratio and the 90% confidence intervals were within 80 to 125 limits is a much more difficult thing to demonstrate, hence the large number of active treated subjects, less placebos. When we sample sized this, no studies had been conducted of this type. So we went to the literature and we selected from the literature data which we thought would give us estimates of variability for trough and FEV1 and for the area under the effect curve for FEV1 on day one. And the sample sizing kind of characteristics are up there. And what we saw was that we were requiring a sample size of around about, I think that says 900, my eyesight's not so good, to allow randomization with 10% dropout from the per protocol set being an expectation of what we might see within the study. Because this was the first study, though, we decided to include a blinded sample size re-estimation. And that occurred when 300 subjects had completed week 4, the day 28 assessment. And what we actually observed was a variability, which particularly for the trough FEV1 was higher than we had expected. And therefore, we'd set a maximum sample size within the protocol of 1,100 subjects, which was giving you about just on 450 for each of the active treatment groups, 90 for placebo. And that was our sample size that we went to for the study. The good news was that the vast majority of subjects made it through all treatment. There were a very small number of discontinuations, well below the 10% that we'd included in the sample size, which gave us a few more subjects getting to the end of the study than we had expected, which was probably helpful. The vast majority of the subjects made it to the per protocol analysis. The per protocol analysis was subjects who had endpoint data for at least one of the co-primary endpoints. And then we looked at sensitivity analyses as well, looking at subjects who provided for both. One thing I didn't mention was the number of subjects which we had to screen to get our 1,100 was over 2,000 subjects. This is not a small study. Our data, though, was as good as we could have hoped, I think. And this graph just shows FEV1 at the bottom, the placebo effect, 0 to 12 hours on day 1 as the beginning of that graph, and to the far right-hand side, the day 28 placebo effect, and then above that, Wixella and Advair largely overlapping. Unsurprisingly, we were highly statistically significant for the assay sensitivity analysis, and our geometric mean ratios and 90% confidence intervals fell within the 80 to 125% acceptance criteria. We did measure adverse events, and obviously in much the way that was described earlier on, we looked at treatment discontinuations as well. And we were pleased that we saw lower numbers of treatment discontinuations in both active treatment groups versus placebo, and also the kind of local side effects that you might expect from an inhaled corticosteroid in this type of study, so oral candidiasis, dysphonia, were pretty much evenly distributed between the two actives and not present in the placebo group. Obviously, the point is, I think, in this type of study, It's a relatively short duration. It's probably not the study to be looking at long-term safety, but thankfully a lot of that is provided by pharmacokinetic bioequivalence, I believe, that shows comparable systemic safety. So we had a successful study on our hands, and obviously that took the product into registration together with all of the other elements of the weight of evidence approach and into a filing with FDA. So the strength, and honestly, I can't overstate this. I mean, I felt we had, without a lot of the framework, which is now being put in place for GDUFA 3, we felt we had great collaboration with the agency to get us to a place where we had a feasible pathway to get registration of this product, a pathway which we just didn't feel necessarily existed previously. The weakness, well, it's always great to be the first, but you also learn a lot along the way, and we were the first, and the first, I think, probably to file with a kind of clinical endpoint bioequivalent study of this size and nature. And what do I mean by that? I mean, it was large, complex, and costly multi-center study. You know, I come from an innovator background, so large and complex studies, something within my experience, but the costly nature of this type of study is somewhere in the region of $30 to $50 million to be able to execute this type of study. And we needed over 1,000 subjects. We screened double that amount. That meant we needed 101 centers in the US. We had competitive recruitment because you don't know how, you know, some centers recruit a lot, some recruit very little. We had a large multinational CRO who were experienced in respiratory studies who ran this on behalf of Viatris. We had to keep very clear oversight of them as we went through the process. We had a lot of the characteristics, central randomization and drug allocation to be able to coordinate clinical supplies between 101 study centers when you're not quite sure who's going to recruit how many is a very complicated process. And this sort of thing is kind of run of the mill for innovator phase 2, phase 3 studies, much less so in the kind of generics area. We did have centralized spirometry, and so we had overread, we had the same equipment at every site, we had the same training applied to the technicians at every site, and with that it took us 9 months to execute the study, which I thought was pretty good. On this basis, it wasn't a typical ANDA clinical study at that time. I think it's becoming more typical now. And obviously, those kind of studies are much more single center, a smaller number of sites, often inpatient and highly controlled. And they have predictable performance and relatively simple execution, although my clinical operations colleagues would probably tell me I'm misguided there. So learnings relating to execution. I think it's fair to say that the agency review of the clinical endpoint bioequivalence study, probably to some degree because of the staffing required within the agency to start looking at this type of study, only really got deeply addressed after our first complete response, which was a major, was addressed. We responded to a major complete response. And that just meant that we were 2 years or more after the original ANDA submission, because we were in the first phase of GDUFA. We were 3 years at least after the clinical phase of the study. Most of the information requests we got from the agency related to protocol deviations and which subjects were included or not included in the per protocol set. And we needed to go back to individual subject data, which we could do, and explain our thinking. The agency inspection focused on multiple individual clinical investigator sites. But the CRO wasn't inspected, and nor was a centralised spirometry vendor, or indeed our sponsor study files. And I think it's fair to say that in this type of study, the usual way of inspection for ANDA studies conducted at a small number of sites, it's hard to reconstruct a large multi-centre study by looking at individual sites. We didn't get many findings, but where we did get findings, there was an issue around retention sample selection. You have to retain samples from shipments of product to sites. Has to be done in a blinded fashion. So the site isn't unblinded, but they need to retain samples which represent the 3 different products which they were receiving. And so that was a complicated process, more complicated than the guidance really allowed for. But we saw our way through registration. In terms of learnings relating to design, from my perspective, it's hard to see how a more efficient study could be designed if equivalence is the target and these endpoints are the target. What I didn't say was that the variability we observed when we looked at the whole data set from the study was even higher again than what we had observed at the blinded sample size review, which meant if you were looking at running this study again in the future, actually you would be looking to recruit even more subjects into the study than we had recruited into the study to have the appropriate levels of power. I'm very pleased to see that these numbers tie in very nicely with the numbers which were presented earlier on in the last presentation. I'm not sure that ours was a selected study, but so we're both getting to the same place in terms of this is a large, complex study if you go down this route. So what about a different design? Crossover studies are something that can be done, more so for bronchodilators than for products containing anti-inflammatory treatments. And so I asked my statistician to look at, so what would this study look like if it were a crossover study? And the truth is that whilst the overall number of subjects goes down, the number of visits remains almost identical. And so what you gain in terms of a lower number of subjects, you lose in terms of the higher complexity of a crossover study design and the inherent problems that crossovers can bring in terms of carryover, washout, stability, keeping patients in studies when everyone within such a study would be subjected to a placebo period, even though only a very small number would need that placebo period for the study to have its power. The only thing which really affects sample size, and that could be applied to crossover designs or parallel group designs, is the acceptance limits. And if you could apply 67 to 150 limits, then obviously your sample size comes down significantly for either approach. Still, in the parallel group design, quite a large study, and obviously it would require comfort around the wider confidence intervals, limits, acceptance criteria. So my final 2 slides, you'll be pleased to know. Surely those leading timing, I'm very pleased to know. In terms of the current clinical endpoint bioequivalence study in the product-specific recommendations, I was trying to think, well, what questions could I ask that would help us in terms of developing these products? And I guess at the moment, the product-specific guidances are just that. There's one for each product. And the question arises, what happens when you bring multiple products along as generic equivalents of reference products where the APIs are different combinations or monosubstances, and the devices, the test and reference devices, are the same between different products? Would you have to apply? And so the examples that I wrote up there was one where Viatris now turns up with FP in hub? Do we have to run another clinical endpoint study for that product? Or could we take pharmacokinetic bioequivalence for that product, plus the clinical endpoint study, which has already shown comparable delivery of the in-hub and discus product of FP and salmeterol as being our weight of evidence approach for the next product coming along? And the same is true, obviously, thinking ahead to the time of developing generics towards Trelegy and Anoro, Brio, Incruz, all being within the Ellipta device. Would you have to do 4 clinical endpoint bioequivalence studies for each of those different products? Or would one covering the triple combo be enough, and then pharmacokinetic bioequivalence for all strengths for the other products? So this is sort of using the current guidance as it is, knowing that it takes time to develop alternatives, and trying to find a pathway which would make some of these products a little more palatable to take forward. And then finally, on the current guidance, what is it that makes 80 to 125 rather than 67 to 150 acceptance criteria? And I don't know the answer to this, but all I do know is that if you're conducting pharmacokinetic bioequivalence studies and you see high variability, you can scale your bioequivalence acceptance criteria based on that high variability. And yet, at this point, we don't have the same facility for this type of design. So would that be a possibility for the future? And then really, for future iterations of the FDA weight of evidence approach to OIP, I guess I have to ask the million-dollar question, which is a clinical endpoint bioequivalence study a requirement for a product where you have shown pharmacokinetic bioequivalence and also in vitro equivalence, and particularly the pharmacokinetic bioequivalence is just exceptionally sensitive to product differences from our experience. And why do I say that? In the left-hand panel here, there's some data that was presented by Sandoz Oriel, which showed products, Advair discus products sourced from the market at the same time, which did not meet pharmacokinetic bioequivalence with each other in what appeared to me to be a well-designed study. We have similar data in-house at Viatris, which found the same thing. There are pharmacokinetic differences which you can detect between batches of reference products in the US and Europe, for example. We did an enormous amount of work, and that central panel is looking at FP pharmacokinetics. Each dot in there and a confidence interval was a single test reference comparison, and the different color coding there, different exploratory PK studies, and we were able to move around the relative exposure of our product versus the reference product by quite small changes in product characteristics in vitro. These really are exquisitely sensitive methods for determining the similarity or difference of products. And whilst I accept the fact that they're not from the compartment that feeds the effect, they are from a compartment which reflects clearance from the site of effect, if designed correctly, which is, of course, locally in the lungs. And, to be able to pass the pharmacokinetic bioequivalence, you really have got to have products which are incredibly similar to one another. And just in the far right there, I just show the results for our definitive study for the 150 product strength of Advair. And I'm aware of the fact that other companies have struggled to do this. And yet, I suspect, probably hold successful clinical endpoint bioequivalence studies. So given the investment required for this type of study, I have to question how much it really provides weight. And so I guess the other question maybe to ask about the guidance as it stands at the moment is just showing test superiority versus placebo enough? It's what was done for the innovator products that were out there. Obviously, it's the equivalence and 8125 limits which lead to the very large sample size. Again, open questions and interested in answers. So, the great news, we've got a feasible pathway. I don't believe there's likely to be any pathway to more sensitive pharmacodynamic endpoints to be able to approve products containing inhaled corticosteroids. I just don't believe that the markers that are available and the long list of them, potential markers has been listed, are sensitive enough when the lowest approved doses of the products are already towards the top of the dose response and on the plateau phase. They are very large studies, the clinical endpoint BIE studies we have, and arguably a greater burden on us as generic companies than on some of the innovator products for registration of the products initially. And obviously, I reflect the fact that the agency got a considerable amount of data when they accepted our ANDA filing for Wixela Hub. And so maybe in the short term, consider areas where we could facilitate these studies within the current constructs, and then most importantly, get around to implementing some of that combination perhaps of pharmacokinetic information and in vitro, in silico modeling information that could allow us to have a new pathway to approval for these drugs in the future.
Okay, my name is Dr. Michael Spagnola, and I will be moderating this panel. I want to thank all of the speakers for those great talks and encourage anyone to submit a question. If you have a question for any of the speakers, please indicate which speaker, if applicable, that you are directing the question to. Don't be shy. And we've already gotten some questions, so I'm going to start with them. Dr. Ward made a lot of good points and some questions at the end of his presentation and some thought-provoking questions. So I'm going to start with one of those directed towards FDA. And so could bioequivalence be concluded using a wider 67 to 150% acceptance criteria given the high coefficient of variations?
Okay. Yeah, so I know there's, excuse me, there's been a lot of discussion about that. And at this point, the product-specific guidance does reflect our current thinking on establishing bioequivalence. But, as we've seen, things can change over time. So just keep an eye out for those guidances.
Okay, thank you. This question is for Dr. Ren. Can the applicant file the bio-IND after completion of the pilot bronchoprovocation study and before initiating the pivotal bronchoprovocation study?
Okay, thank you for the question. So the requirement of submitting the bio-IND is the same for the pilot study and the pivotal study, since the methylclonin concentration used maybe exceed FDA recommended the amount. Also, if the PD study is conducted outside of the US, based on the CFR 320, 120, the FDA maybe can accept the well-designed, well-conducted with non-submission the IND study if the FDA will validate the data through the inspection. Thank you.
Thank you for your answer. This one is for Dr. Ward. Despite the large cost and large number of study subjects needed for the comparative clinical endpoint and PD bioequivalence studies, sponsors have had greater success conducting and submitting these acceptable ANDAs. Why do you think that's the case, and what are the biggest obstacles to successfully demonstrating bioequivalence using alternative methods?
Well, I'll take the first part of that question. I suspect that the workshop is going to try and answer the second part. So I think the reason that we don't have alternatives yet is because certainly for quite some time we were focused on using the pharmacodynamic endpoints as markers, as showing dose response for these particular active moieties, and going through the not insubstantial investment scientific learning process to look at those different markers and find out that unfortunately they didn't show the dose response that we'd all been hoping they had. I think then when the OIP product-specific guidance came forward, we were just so delighted that there was a pathway which we felt that we could follow, that we were happy that that had been defined, and so we got on with following it diligently, and with a lot of the checks and balances for that type of study being put in place to ensure we came out with the best quality study that we could to try and help our kind of review. I think it's only now, as we look back and have 10 more years of experience, that we kind of see just how big an investment these are. And whilst they're, as has been described earlier, for the real blockbuster products, they are investments that companies are prepared to make. I think we're now looking at what about for the smaller products where those investments perhaps don't make sense, and yet they're important products where we would like to improve access for patients. How are we going to go about doing that? And also, I just believe that the whole science has moved on by 10 years. Perhaps some of the biggest barriers perhaps remain the fact that you guys at the agency see all of our joint efforts around the products that we've sent you so far. We only see our individual contributions. And some of this learning that can then be applied to develop alternatives really requires access to probably data from multiple companies with multiple assets to be able to move on the state. So that's where I see this, and again, hearing the opening remarks today as being, a fantastic way in which to be able to facilitate that happening, to a point where I hope within the next 5 years, perhaps, we're starting to shift the paradigm. It took us 20 to get to the paradigm that we had. If in 5 years' time we could be shifted, we have today, if we can shift it again in 5 years' time to something simpler, I think that would be fantastic. But it's going to be a joint effort, truthfully.
Thank you for sharing your thoughts. And obviously, this workshop is helping to drive and remove some of those obstacles. This question is for Dr. Gong. What could be potential alternative approaches, alternative BE metrics, to address the challenges in a clinical endpoint study?
I think today in my talk and Dr. Warren's talk, so we have shown there are a very high set variabilities for the FEV1 matrix. So I think part of those variabilities came from because the study is conducting a more variable patient population. So one of the potential option is the one I presented, use other analysis model covariance adjustment to trying to reduce variabilities. And of course, if alternative PD models can be explored to have a more sensitive or less variable matrix over the FEV1 matrix, and I think that can be the alternative ways also in the future. Thank you.
Thank you for the answer. I'm going to turn to a question for Dr. Ren that we've received online from our virtual audience. We received several questions about different types of nebulizers used in the bronchoprovocation test. And this one is, methacholine in a bronchoprovocation test is delivered by nebulization, but there are many different nebulizers that are used and by different methods, continuous versus single breath. What approach is recommended by the FDA in order to decrease the heterogeneity between different investigator studies and importantly, also like-for-like comparisons between different studies and investigators?
Thank you for the question. So we don't have specify what kind of device you should use to nebulizer to deliver the methylcloning. But the dose of the delivered methylcloning can be calculated based on the device specification. And since the study was a test and reference was conducted at the same condition, so this potential difference will be very minimal. Thank you.
Thank you for answering that question. The next one is also from a virtual audience. It's for Dr. Soukup. With respect to treatment failure, as you say, this may be due to subject behavior, specifically behavior in inhaling from a DPI. How do you ensure the patient is consistent in their inhalation flow and technique in their inhalation over the 28 days in the study, as inconsistencies will be a critical co-founder in the data contributing to treatment failure?
It's a good question, and it's definitely a difficult piece. For that, you just have to really focus on your counseling of how to appropriately use a DPI, and we would recommend a good, solid, consistent instruction across all sites, just so each subject receives the same training, even if they're familiar with the product, just as a refresher.
Thank you for your answer. This one is for Dr. Ward from a virtual member. For DPIs, device resistance is considered one of the significant parameters. Please share your thoughts on device resistance parameter as a weight of evidence for ANDA application.
I think that device resistance is definitely something that can be measured. And I'm sure that if there were big differences between test and reference products, then there could be issues. Whether those issues could be overcome by other elements within the formulation, I think that's certainly a possibility. But I think the idea that test and reference devices should have comparable device resistance, that you shouldn't be comparing a very low test device with a resistance, very low resistance test device with a high resistance reference device makes perfect sense. You shouldn't be going down that pathway, so try and get them comparable in the first place. I don't believe, though, I'd say this off some experience, that it is a critical factor to absolutely match, as we don't for Wixella InHub, and we've shown that and published on it, so it doesn't have to be completely matched. But probably, were we starting again, we would have kept them identical from the very beginning, truthfully. I just have a couple of other comments as well about the methacholine challenge, because I mentioned it we were looking at it for salmeterol. We were somewhat shocked when we came to run that study, when we realized that much of the guidance which is out there around how to conduct methacholine challenges was on the basis of a mini Wright nebulizer, I think, which had been discontinued everywhere and didn't exist anymore. And we found that with new nebulizers, you were looking at quite different performance characteristics, much more efficient nebulizer systems. And I think that speaks to making sure that you do your pilot study correctly if you're going to implement this in methacholine challenge, for example, for albuterol nowadays. We were finding delivery times which were way shorter than they had been previously to get you to a comparable lung dose. So you need to look at these things carefully. As with all of these studies, everything moves on over time, and everything is about diligence and making sure that the study you're doing at that time has been as diligent as possible in its science.
Thank you. Do we have any in-person questions? Go ahead.
Hi, this is a question. Oh, sorry, that's loud. This is Andrew Cleveland from FDA. I have a question for Dr. Ward. I was just hoping you could comment on the potential impact of either pragmatic or adaptive designs in terms of feasibility and cost for such a comparative clinical endpoint trial, as well as if there's been any discussion of having some sort of platform trial to test multiple of products like this in a kind of sequential way.
So the second question first, because that's easiest. To my knowledge, no platform trials have been talked about so far. I guess in terms of the kind of pragmatic design, and I don't think anyone's gone down that path. I mean, we were very pleased that we, in terms of adaptive designs, we were very pleased that we'd at least included a blinded sample size re-estimation. I guess the problem with adaptive designs is that you take a hit in terms of power if you're looking at data in the middle of a study. So I don't think I'd want to do that in your definitive clinical endpoint bioequivalence study, because we would end up having to recruit even more subjects to maintain the power of the study. But is it something which could be considered? I mean, these things can always be considered. I'm just not sure how it would necessarily help us, because I think the variability is kind of inherent in the measurement and the patients and the duration of the study. It's obvious when you look at much lower variability for the day 1 endpoint than for the week 4 endpoint. There's something about how it's being captured over a time period within a patient population, which just leads to variability, which I'm not sure you're going to get out of the measurement. So I'm not sure that looking at the data as you go through is going to help a great deal in that respect.
Thank you. Next audience question.
Hello, good afternoon, good morning. Question is for Dr. Ward. And before the question, I have 3 very minor quick comments. Your presentation does underscore the need to get rid of these clinical studies, which cost, when you did this study and some other sponsor did, it's about $40 million. And if it doesn't tell us anything, whether you give 25% drug less or 25% more, the response is going to be the same. And we have done some pilot studies and other things, and some of the other things you mentioned about ENO and all that, I was at the agency when those were initiated, or some other studies for albuterol that were done, but nonetheless, it does. And second is the question about 67 to 150%. That dose scale procedure was invented by the agency in mid-90s for the approval of the first CFC albuterol. And it is specific only to the studies which have dose response. And what were the reasons? I can't go through that, why it was invented, how it was invented, but that it has never been applied anywhere else. But within the agency, there is another example of widening the confidence intervals on dermatological corticosteroids. It is 70 to 143%. But there also, there is a pilot study which is done to determine the dose response, and based on the pilot study, you choose the dose. So there again, it is within the, what to call rising part of the curve. Then within the study, you have lower and higher dose to determine whether the actual test and reference one dose is within the rising part of the curve or not. And then I personally believe that it will be hard for the study for agency to widen the confidence intervals, because there are 3-4 successful studies submitted to the agency already on Advair and some other products. Then I have one question. Your presentation concludes very well, and I think it's very true that PK is the most sensitive marker. But now the in vitro studies that we do, the clinically relevant in vitro testing and all that, and some of the other things recommended in the non-ABE are all even more sensitive, right? So whether there's any difference in the 2 products, that is where the opportunity is. For the PK, I think we go as back as 2009, when GSK published a paper, remember that Peter Daly Yates paper, which was actually published against the European guidance, which recommended PK. But their own paper showed that PK was more sensitive than in vitro, whereas the clinical study showed those products to be bioequivalent. PK showed they were 200% difference. So given that, let me ask you a question about the PK studies that your team did for Vaxilla. Correct me if I'm wrong, you used 3 puffs, 3 actuation doses, right?
Sorry, yes, 3.
3 actuation doses.
That's right.
Was that subject to a bio-IND in the US or not?
It was, yeah.
Okay. If we look at clinically, and also Mylan published a paper which I read carefully, and the reason for 3 actuation given was to reduce variability, which in my opinion, the agency does not allow. If the agency knows that this is done to reduce variability. The reason is that clinically, if you look at when there is a bronchodilator involved, and we saw that in some of the Johns Hopkins studies when were done for albuterol, the deposition of the second puff is different because the first puff opens the lung. And then when you go to the third puff, most likely the product differences are going to go away. So the question that I have is that inability of Sandoz to have an successful application failed because of the PK only. They had successful in vitro, successful in vivo. If Mylan has done the study as recommended in the guidance for the single puff, because at the time Mylan did these studies, very sensitive assays were available, and I'm sure you employed the sensitive assay. Would you be in the same boat as the Sandoz?
So if I remember the guidance correctly, and it's been a while ago now, it suggested that the number of puffs used, the number of inhalations used, was the minimum number required to be able to determine robustly a PK profile. And that work actually was conducted prior to the Mylan days within Pfizer to look at 1 inhalation versus 3 inhalations of the reference product. And it wasn't just about variability, it was about detectability over time. And that was using assays which were 1 picogram per mil assays. These are hard drugs to track. And you see that even today in the innovator products coming into the market, umeclidinium can be tracked for, 6 hours? I think we determined that going down the 1 inhalation route actually wasn't the right way to go to be able to look at the product. And we showed the variability we looked at within product variability for the reference product. We weren't looking at test versus reference, we were looking at the reference product itself. And of course, coming back to your point about scaling of bioequivalence limits, if a product is highly variable, you get to scale the pharmacokinetic bioequivalence limits accordingly based on the variability.
Yeah,
and I guess we were trying to avoid that by going in with a number of inhalations, which we thought was justifiable. In terms of would we have suffered the same fate as Sandoz? I have no idea. Because we haven't looked at that, gone down that route. But I think the whole point of the weight of evidence approach as it currently sits is that no one piece of evidence is the be all and end all. It's the sum total of that weight.
First of all, I congratulate you on your success. But my concern is that the variation in the standards that agency might apply in one case 3 puffs, in the other case people are struggling with 1 puff. And I hope that does not happen in the future. Because even at the time Mylan did this assay, in my experience with 1 picogram, you can go up to 5 half-lives of salmeterol. And even the lowest fluticasone, and people have done it very well. It is highly variable product Advair, no doubt. And the people have tried various models. We even tried going to replicate design and all that. But to my knowledge, the agency allows more than 1 actuation based on the sensitivity of the assay, not the variability in the response. So therefore, I think this is a general comment, both for the agency and everybody, that, okay, what is done, then in future we should look at it. And same thing has happened long time ago in one of the nasal sprays, where somebody did more than one, got approval, then somebody came with one perfect, beautiful study, which I remember. And then we were looking backwards and saying, oh my God, this. But nonetheless, I think given Sandoz situation, and also given, which I know that as you go from 500 to 100 microgram strength of Advair, your possibility of passing the PK studies goes lower and lower. At 100, you have the least possibility because of whatever reason. And the sample size can go from, let's say, 60 to 150 or more. And in all the 3 studies Mylan did, I was surprised that they used 66 subjects, regardless of the strength. So in my opinion, there is something that indicates that there was something not right over there. I'm not saying to go back, but I hope that agency in the future looks very carefully at such a thing. Thank you.
I think I would just sort of add to that point that in our experience, the 100 microgram strength was the most difficult to get equivalence for, full stop. There was understanding you needed about that particular strength, which was more evident at that strength than any other as well. I'm not sure that the 3 inhalation versus 1 inhalation thing was the key to that. I'm really not. But it would require studies to look at that. And what I would say is that when we started out with that low strength and doing the methodology study that we did because we were concerned about that.
My only point is when I say consideration of the agency, we have to look at what is allowed in clinical practice. And that has to be reflected in all the evaluations we do. So in the clinical practice, the highest dose is 1 puff. And we also know that when there's a bronchodilator involved, and it takes about 5 minutes to give 3 puffs, the deposition for the second and the third will be very different. Thank you.
There you go. I mean, he's completely right. This should never have been accepted. Because in this product, there's 2 API. One has a Cmax that has occurred very fast. The other one is delayed. So the one that is delayed, it's not a question. But if you give 3 puffs and it takes 5 minutes, it becomes kind of a continuous infusion of the dose. It becomes way, way, way easier to pass.
I mean, how shameless to be saying that, you know?
So you're completely right.
I would say that, number one, it doesn't take 5 minutes to take 3 inhalations from device. It's probably more like 1.5 minutes. I think it's also fair to say that these studies were conducted in healthy volunteers, in which very little bronchodilator response would be expected. Again, I think we have to be open to ways of making these products amenable to being put into studies and being looked at. And personally, I don't see the problem with having to take more than 1 puff if necessary, provided you show that it is necessary and that you're not doing it for alternative reasons. And I think we did that. I think the agency saw that data and they accepted it.
Xianmin Zhen from Transpire Bio. I have a question for Dr. Ward. And you mentioned that you have had experience with 2 clinical endpoint studies. So my question to you is that based on your experience, did the clinical endpoint study even add any value whatsoever to the totality of the evidence for bioequivalence? So if you remove the 2 clinical endpoint studies from your package, do you think you already have generated enough information to demonstrate bioequivalence based on in vitro and PK or other studies? Thank you.
Okay, so I think 2 ways of answering that. Number one, if I was developing the product in Europe, then the agency would say, yes, you've got enough if you've shown pharmacokinetic bioequivalence. I don't believe that the clinical endpoint bioequivalence studies, and I think it's already mentioned already, Peter Daly-Yates, Dave Parkin, looking at the ARPID device versus the discus, PK 200% difference, and yet the conclusion was I never really saw whether it was a rigorous determination of clinical endpoint bioequivalence. My suspicion is not, but the efficacy looked the same, was the kind of conclusion. Which says to me that it's quite easy for efficacy to look the same when products are really quite different. So I think for that reason, I believe it adds something in terms of just demonstrating to everyone's satisfaction as best as you can without more sensitive methods being available. Yes, you've got the same activity within the lung. Do I personally feel like that is worth the investment that is required? I'm not sure that showing equivalence versus just showing superiority versus placebo, is warrants that additional expenditure. And I'm pretty certain that that currently it is holding back more people from being involved in this area, developing and delivering products which are generic equivalents of the reference products that are out there. I'm pretty certain it's holding people back. So if you're European, you would say you don't need it. But I understand why it's there in this context. And I just hope that alternatives are delivered soon to everyone's satisfaction.
Thank you. We have another question from the audience.
Leslie Hendless, Gainesville, Florida, for Dr. Ward. Practical question. The Wixilla has to be administered in a vertical position, and the Advair discus is in a horizontal position. I find that patients in our clinic don't know that. The pharmacists don't tell them that. And my question to you is there any data indicating that by taking the Wixilla in a horizontal position, you're getting less drug delivered to the airways?
Okay, thank you. That's a good question. And I would say that it was a significant part of the agency's review of the product because the difference was obvious from the very first days. We certainly have the in vitro data to show that the product delivers equivalently. And believe me, it wasn't just done in the 2 orientations. It was actually done, I think, in 16 different orientations, taking it between every single way, and showed that the product delivers equivalently regardless. I was discussing with someone earlier on as well that in clinical practice, you quite often see people using a discus inhaler in different orientations as well. And so you're against the backdrop of how the reference product's always been used as well. We also did some patient experience human factors studies, which showed actually the number of patients who tried to inhale, just being given no instruction, but being given the drug in the commercial packaging with the instruction leaflet, tried to inhale in the wrong orientation, was relatively low. And in those subjects who did, we also conducted immediately following that flow profiling studies to understand how they got a lip seal around the device in that orientation, and did they get adequate flow through the device, and found that all but one subject did. And that subject simply had such low flows that they probably wouldn't have got the dose from a discus either. It was an issue that we saw right from the very beginning. It was certainly tackled in the review of the product, and I think we show that the devices do deliver equivalently.
Thank you. We have another question from the audience.
Hi.
I just have a follow-up question. Do you have any post-marketing data with related to the device orientation? Have you collected any post-marketing data and show the compliance of the patient use of these two, different oriented product? I think there was a big discussion at the time when we review and approve this product. The device similarity was a big discussion. We saw the data pre-approval, but my question is, do you receive any or collect any data post-approval to suggest the exchangeability of these two products after it is put on the market?
A couple of answers. Number one, I'm learning that if you're an industry person, you should probably make sure you're balanced on the top table, or I should take all the questions. Number two, I don't think that we have conducted any kind of post-marketing surveillance of that particular aspect beyond the post-marketing surveillance that would be in place for device complaints or adverse event reporting, that kind of thing. So no, we don't have any additional data beyond that was presented for the registration.
I'll add just that FDA also performs pharmacovigilance, and so in addition to Viatris, FDA also evaluates after post-marketing. So I think just looking at time, we had time for one more audience question.
Hi, it's Yuqing from Apotex. Question to Dr. Gong. In your presentation, you have looked at the use of covariate analysis to reduce variability. Unfortunately, you show that only reduced variability by 10%, I think. Now, you didn't identify which demographic factor that gave you that 10% reduction. But nevertheless, the reduction is relatively small. But it's not surprising that because the major source of variability perhaps was due to intra-subject variability. With that, I can understand why the intra-subject variability can be high, because your trough FEV1 measurement is based on one data point. That usually gives you very high variability between subjects. Have you looked at the use of perhaps repeated measure mixed effect model, utilizing the data, utilizing FEV1 data that you collect from week 1, 2, and 3, and 4, and use those measurements to do your repeated measure analysis, or maybe even calculate the area under the effect curve based on those 4 measurements. That perhaps could help you to reduce the intra-subject variability significantly. I don't know if you have tried that or not.
Thank you. I think this is a good question. As you mentioned, they are for our study, we showed only maybe a limited portion, like 10% subject can be reduced by applying those covariates. So part of reason is those covariates we are looking at the baseline factor, like those don't. So there are more factors can cause higher variabilities. So for our study, regarding your question, if we look at different periods, so for this results I presented, it's from the data that's submitted to the FDA. So we only have the data that collected for those endpoints, for the drug approval. So we did not have, we did not analyze those data, like you said, to further to look at the variabilities, how it came from.
Well, as I said, looking at the baseline data will only help you to reduce the inter-subject variability. But for intra-subject variability, you need to look at repeated measures type of analysis.
Yeah.
Just picking up very quickly on that. It is not a single measure of FEV1 that was done at day 28. There are 2 measures, they're half an hour apart, but they need to reflect the trough measure that you're getting. Obviously, I think there's 2 elements that reduce the variability of the AUEC 0-12. One is that it's 0-12, but that is reflecting probably the salmeterol components first dose. And if you did that again on week 28, you'd be looking again perhaps at the salmeterol component rather than the fluticasone. And also, I think, the main component is that the baseline is measured immediately before you run that AUEC 0-12. Whereas in trough data, by definition, the baseline has been measured 4 weeks previously. So we have looked at repeated measures, not beyond 2 or 3, I think, maybe maximum. But we've implemented 2 to try and bring variability down a little. It doesn't help much.
Thank you for the question and answer. Join me in thanking our speakers once again for their answers and presentations. And that concludes the panel discussion.